Good morning, everyone. Today we will start with Activity 3, the structure and the chemical components of chromosomes. We will talk about the components and the constituent of a chromosome. In a cell, we can find the nucleus, the mitochondria, the organelle responsible for the energy, and other parts. The nucleus is the main part. It has the chromosomes. As we've said, in a diploid cell, there are 46 chromosomes, while in a haploid one, there are 23 chromosomes. A chromosome is the major part in the nucleus that constitutes the genetic information. It means like eye color or skin or any other trait in a human being. Okay, so in the chromosome we have the genetic material. How is this genetic material arranged? We have a structure called DNA. DNA or deoxyribonucleic acid. It is the major part that carries the genetic information. It means represented by genes. As we all know, we have genes for every trait in our body. So DNA is present inside the chromosome, as we can see in this structure. It is a double helix. It means it is doubled or like a ladder. In the chromosome, we can find the DNA, which is responsible for all the genetic information and all the genes. DNA identification by full-gene reaction. How was the DNA identified in the cell? First, to identify DNA, a stain was done. A stain, it means coloration or staining. Cells were stained with full-gene stain. It is a reagent that colors the DNA with red. When applying this stain, it was found that red coloration appears inside the nucleus of a cell. Thus, they concluded that the DNA is present inside the nucleus of a cell, and the DNA is colored, not the proteins. So, identification of DNA was done by full-gene reaction. We can see here the chromosomes are colored in red because the DNA is found in the chromosomes inside the nucleus. And we all know in somatic or body cells, 46 chromosomes are present, while in gametes it is the half or 23. So the amount of DNA is half in the gametes. Thus, DNA is identified in the nucleus and on the chromosomes specifically. From chromosome to chromatin and vice versa. We want to know the changes or inside the chromosome what's happening when moving from chromosome to a chromatid or chromatin. It means from mitosis to interphase or from interphase to mitosis. First, starting with a chromosome at mitosis here, it is composed of two chromatids joined by centromere. If we get this part of chromosome or inside a chromatid, we found this condensed form of the chromosome that is composed of DNA. And this is the extended form or chromatin. It is long and thin. This will be in the interface. In the chromatin, we can find the DNA, and the DNA has this structure. It is like beads on a string. We can see that we have a nucleosome. A nucleosome is DNA wrapped around a protein called histone. It means this is a protein and this is a protein and this one. The protein is called histone. The DNA, this is like a thread. Okay, it is wrapped around this protein. We can see here the DNA molecule is not wrapped around any protein. In some parts it is wrapped. The part where it is wrapped around the protein is called the nucleosome. This is the beads on a string. Okay, and this is called the condensation. From a chromatin to chromosome, there is condensation from interphase to mitosis. The opposite, from mitosis to interphase, it is called decondensation. It means the chromosome was of two chromatids becomes of one chromatid, and it is called chromatin. It is long and thin. Okay, this DNA structure, you should know that where the DNA is wrapped around a protein called histone, this part is called a nucleosome, and in some part the DNA is not wrapped around any protein. This is a DNA structure. It is a double helix or a ladder form. In the chromosome, we have two chromatids, and in each chromatid, we have a double helix DNA. The DNA is a large molecule or macromolecule. We take here a part of the DNA, and we want to know the components of this DNA. The DNA has many nucleotides. The nucleotide has three components: phosphoric acid, represented by P, deoxyribose sugar, represented by D, and a nitrogenous base, represented by A, C, or G, or T. It means we have either A or C or G or T in the place of nitrogenous base. A stands for adenine, C cytosine, G guanine, and T thymine. It means the DNA has many nucleotides, and every nucleotide has three parts: phosphoric acid, deoxyribose sugar, and one of the nitrogenous bases. Okay, in every strand of the DNA we have this structure. It means a nucleotide bound with another nucleotide, and as follows it continues. We can see here the arrangement of the chemical components of the DNA. I have phosphoric acid, deoxyribose, and here G, guanine. Then we continue on this strand: phosphoric acid, deoxyribose, then cytosine, and we continue. It is a large molecule. On the facing strand, we have also phosphoric acid, deoxyribose sugar, and we have a base from the four bases, but you should know that G pairs with C, forming a base pair. Here C with a G by hydrogen bond. But T pairs with A. This is also by hydrogen bond. So we have two base pairs, two types, GC or TA. Always G pairs with C, and C with a G, and T with A, or A with T. So I have two strands facing each other. They have hydrogen bonds between them, and they are composed of many nucleotides.